that 5 o'clock UK time, so I reckon we'll get started. So welcome to our Neo4j online meetup. So today we're going to have a talk with Nikolas about Pheno4J, a gene to phenotype graph database. So I guess I'll get started. So in case you've not joined us for any of our previous meetups, my name's Mark Needham. I work in the developer relations team at Neo, and with me you have Nikolas. So Nikolas, I'll let you introduce yourself. You'll have to unmute.
Hi everyone. Yep, can you hear me now?
Yep, we're good.
Cool, great. Hi, thanks for the introduction, Mark.
So do you want to give maybe just a quick introduction to yourself as well? I guess, yeah. What do you do to start with, perhaps?
Sure. Shall I? Yeah. Sorry, there's an echo at the moment because maybe I should close the YouTube channel. Okay. All right. Okay. There you go. So yes, I work in genetics at the Institute of Ophthalmology. So this is the research institute affiliated with Moorfields Eye Hospital, and we work on genetics of eye disorders. So we DNA sequence a lot of patients, and we have a growing database of DNA mutations in various patients with eye disorders. And part of my work is sifting through all these mutations and trying to find out which ones are the ones causing the specific disease. So there's different kinds. So I don't know if any of the participants here know much about DNA sequencing, but basically, when you sequence a human, you will find, depending on the type of sequencing you do, you'll find more than 100,000 genetic mutations. And if you do what we call whole genome sequencing, so if we look at all of the mutations, you'll get up to 4 million. So these are very big numbers, and we were trying to store this in a sort of efficient way, efficient database that we could then search. So we first considered, well, relational databases would have a problem because you'd have many of these, each person would have many of these mutations. So you can imagine there'll be many very large join tables for person to mutation. These are not really appropriate for this type of data. And a friend of mine, Sajid, who's participating here, is suggested we use graph databases. And so he coded up what's Pheno4J. So it's basically using a Neo4j graph database, and it's some Java parsers which read in our data and load it into the graph database. We can point you to the GitHub project.
So I put a link for the GitHub project on there. And so did you try putting the data in any of the relational databases or other databases before you built Pheno4J?
So initially we started with a document database which started with Mongo, which obviously wouldn't let us store all the relationships of mutation to person. So what we were doing is we were only storing for each. We had a mutation as a record, and then we were only storing up to 100 people that had that mutation. So yeah, we couldn't store all the information in there.
Cool. So I reckon now might be a good time for us to maybe we can take a look at the model of the database that you've created.
Sure.
So let me just hand over control to you. So you can share your screen. Let's see with the button.
Okay. Yeah, I'm going to do the screen share. Share. So can you see the picture here of the figure?
Yeah.
Great. Okay, so this is the overview of the database. So as I was describing earlier, each person that we sequence shows up as a node here, and then all their mutations, this is the mutation node. And so these relationships, this is a massive many-to-many relationship here. So each mutation can be seen in many people, and each person will have up to 100,000 or even more mutations. And then each mutation occurs in a gene. So this is this relationship. And yeah, I'll forget about this because we might be going into much biology here, but I can talk about it later. And the other key aspect here is each person has a set of clinical features which describe basically the disease that the person has, and that is the link that you see here. So these features might be seen or might not be seen. And next link, which is also very interesting, is this relationship here, which is the gene to clinical feature. So I can explain to you why it's called an HPO term later, but anyway, just read this as a clinical feature. So this relationship here is what mutations in a gene. So this is what's known about from basically reading publications and so on, what mutation in what gene causes what disease. So this relationship here is loaded from a database of genes to clinical features, basically to diseases. So that's the main nodes in our schema. And next, what you have here is these gray boxes here are the input data files, which we use to load into our database. So the gray boxes, which have a little wavy outline here, are the actual files, and then the square boxes here are the parsers. So this file here is the file of all the known clinical features. So it's called HPO because it stands for the Human Phenotype Ontology. You can just Google that. I'm not going to Google it now, but you could just Google it and you'd see what it's about. It's basically an ontology of clinical features. This here is what I described earlier as the known clinical features to gene relationships. This is downloadable from the OMIM website. Maybe I can put that on the chat here. If you just Google OMIM.
Yeah, I can probably do. I can do that.
You can do it, maybe.
Yeah. So I guess with that model, is the one that's on the screen now? Was that the first version of it? Did you iterate a few times? What was your?
No, we had a few more nodes in there, and then we simplified things a bit. For these two nodes, for instance, are there to reflect the biology, but not that useful in our queries because it's very rare for us to query with these. But maybe in the future these would be more interesting, more used. So I think when Sajid, Sajid is available on the chat, I think maybe you can ask him there. But I think this is pretty much. Yeah, it didn't change that much over time. We maybe had a few more nodes.
Cool. And then on each corner of you've got those are like different integration points, aren't they? Different data sources coming in. So you've got
absolutely
the VCF parser and then the Persian parser. So are those like well-known data sources, or what would they be?
Yeah. So it's described maybe better in the paper we've written, but some of these are user specified, and others are downloadable from websites. So it says here downloaded the HPO file. These two are downloaded, and these two would be provided by the user. The VCF is a known for anyone working in genetics. The VCF is a known, very well known data format for storing genetic mutations. So it's basically a matrix with as columns your individuals and as rows the mutations, and you'll get either a 0, a 1, or a 2 there, depending on if you have how many copies of that mutation you have.
Cool. Okay, so should we?
Yeah. Mhm.
Cool. I reckon that's all I can think of to ask about the model, apart from the fact that I don't. The domain's probably a bit difficult for me.
There's
But maybe
one thing actually, yeah, that I didn't talk about is this bit, which is very important, which is these are the mutations, but mutations are by themselves are just names basically. So you need to know more about where the mutation, what gene the mutation is, and what is its frequency in the general population, and so on, because that gives you some idea of whether it's likely to cause a rare disease or not. And all that information comes from here, which is an annotated set of variants, basically. So we run this tool called a VEP, which is, again, you can just Google that, variant effect predictor, which takes all the variants as input and creates a JSON file, which we then parse and load into our database to create these nodes.
Yeah, and I think all those parsers are in the repository, aren't they? The one that we talked about.
Yeah, so there's Java versions of them, which Sajid has written, and I'm writing some Python versions as well.
Cool. Okay.
And maybe I should mention how we're doing the initial import. We are using the bulk load tool, which Mark can link to, but it's basically for constructing the database from scratch rather than updating it. Am I right?
Yeah, exactly. So I'll put a link to it on the chat. But yeah, that's the tool that basically works. It just builds the database directly. It sort of doesn't bother with creating any transactions or any of that safety stuff. So you tend to only do it once, usually at the beginning of the project, and then if you want to completely replace the database, you could use it then as well. Otherwise, I guess people would be using Cypher in some sort of form to do their updates.
Yeah. Actually, I was thinking.
Sorry, go on.
No, I was thinking one thing that could be nice in this diagram is to have the numbers of here for the relationships, just to give some idea of the magnitude. It is a very, especially here, this relationship is in the order of millions, even more. I can just count the number of lines in the import CSV file, but it's a very large data set, and the bulk importer does it very surprisingly fast.
Cool. So maybe so I guess we're probably not going to go into the details of the import. We basically wrote a custom parsers to get that into the format that the Neo4j import tool likes. But perhaps we can have a look at what it looks like. I don't know. What was your next plan? Maybe have a look at what it looks like once it's imported.
Sure. Yeah. So we can use for that. I guess we can use the interface. So here are the node types that we've got loaded here at the moment, and if I go to say a person, these are my individuals here. So here I'm only looking at 25 of them, but I think there's about 6,000 of them in the database. And just going on one, you can see the attributes here. So each person has an ID, a gender, and then some sort of score, which is basically just describing how good the phenotyping is for that individual. If I click on that, and I haven't played that much with this, but whoa. So these are just a subset of the genetic variants that person has. As I said, here we see rendering was limited because there's many of these relationships. But if you click on one of these variants, then you should be able to see who else has this variant which other person has this variant in the database. So yeah, quickly gets. So this variant is a common one, as you can see in our database. So many people, oh my God, many people have it.
Yeah, your browser's not loving you now.
In blue, what we've got here is a gene, I think. So that variant is, so this is obviously not the way we would be browsing this data. I'll get onto that, but we've written a web server called Phenopolis, which we are using for browsing data. Okay, it's getting a bit busy here.
Maybe try :clear to clear out your browser windows. Cool. So you hit one of the really dense relationship types, I guess.
Yeah, exactly. maybe I should start with maybe a gene would be a more sensible entry point.
So these are, what are these? Are they're representing like one? Is it every single gene that exists, or is it like a subset, or are they which genes are loaded in?
So yeah, every single gene is about 25,000 genes, and every single gene has a number of mutations, which are mutations which are existing in our data set. So we're only showing. So if I do this, it'll only show the mutations. Okay, so this is showing the transcript. That's interesting. I haven't seen this before.
Oh wow, there's a lot of relationships there as well. So that's between. So what's a transcript in this domain?
Okay, I think I understand why. Yeah. So each genetic variant. So this is a bit more complicated to explain this. We have to get into a bit more biology. Maybe I can explain it here. So each genetic variant. So firstly, sorry. I'll start with each gene. Basically, leads to multiple transcripts, and these genetic variants will appear on certain transcripts, but not others. So what we're storing here in the transcript variant is the effect that the DNA mutation has on the transcript. So you have like quite a big. So each genetic variant will have multiple transcript variants, basically. So that might explain this number of relationships here we're seeing here. How would I go and just display the just a genetic variant here? Instead, can I choose? Oh, maybe. I see. Let me just see something here. Right. So if I want to see the genetic variant.
Yeah, maybe it's easier to browse from that side.
Yeah.
Yeah, to narrow it down from here, you would have to write a bit more of a specific query, which I don't know.
Right.
How easy that would be to do.
Can I do it from? Do I have to write the query, or do I have to? Can I do like? Genetic variant like this, and return.
Yeah, to return GV as well.
GV.
Oh yeah, there we go.
Okay,
that's better.
yeah, that's what I wanted. Okay, so yeah, so this we're just showing 25 of the genetic variants in that gene. So if we want to see how many it had, then I'll do a count on how many there are in total, or I don't know if there's a way of doing it for the GUI, but is that right? Okay, well, that's oh yeah, but sorry, that's all. Yeah, sorry, I need to specify the gene. Yeah, where N gene name T span 6.
Is that a good gene?
I don't know. It's just a random gene. It's not my. It's not a very big gene because it doesn't have that many variants in it.
So what does it mean? So that means there's 61 different versions of that one?
That means there's 61 mutations in that gene.
Okay.
Yeah. So
what would be a mutation? What does that mean?
So a gene, I have to show you a picture of what their DNA looks like. But a gene is basically, so a DNA is just a sequence of letters.
Okay.
And we've got 3 billion of these letters in a human genome, and then each gene is just a region of that genome. and then we compare that region to a reference, and then if it's different from a reference, then that counts as a mutation.
Okay. So you kind of pre-process the data to figure out the mutations.
Exactly. Yeah.
Oh,
yeah.
cool.
So 61 mutations means that at 61 places within that region, there are differences to the reference genome. So yeah, some genes are much bigger and have much more mutations. So I think if we try this one, oops. Yeah, so that one has a fair.
Oh wow, that one's got nearly 1,500 different variants.
The biggest one.
So what does it mean? Does it actually mean anything if they have more, or is it just?
So bigger genes obviously have more mutations.
Okay.
But then you're right. Usually genes which don't have many, which are, imagine if a gene is very large and has few mutations, that usually is indicative of it being resistant to mutations. Meaning that if you have mutations in that gene, then you're less likely to. It will be a selective disadvantage. So mutations will be. That the person that has the mutations will be less likely to pass on those mutations to the next generation.
Okay.
Because the human genome has been evolving, and every genome on the planet has been evolving for millions of years, and we've got all this selective pressure, which explains why some mutations are more common and some are selected against. So this gives you.
So what does it mean? What's the base genome? So is that how far back does that go?
A base genome is actually just a agglomerate of, I think, 12 genomes. So the human genome was completely sequenced, I think, finished sequenced in 2001, and they combined DNA from several people, including a person who was called Craig Venter, who was active member of the sequencing project, and so it's not very representative of the whole population. And
okay. Oh, interesting.
Yeah, so that explains why partly we see so many mutations in a typical genome.
Because the base one wasn't necessarily like the most generic. Yeah. That it could have been.
In fact, because of the way human population has evolved and started in Africa, this is where Africans tend to have more mutations, because what happened originally is we all started from a population of say 100 individuals, and then a few migrated and went to populate like other parts of the world, and so the genetic pool was largest in the starting population from Africa. So that's where you see the most genetic diversity and the most mutations. So when you do the sequencing, if you see an individual with a lot of mutations, that typically shows that that person is probably has African ancestry.
Oh, okay.
It's quite cool. Yeah, and it's quite interesting. The reference, and in fact, actually, the reference genome at the moment is just a sequence, but because there can be all these different mutations, and also there can be insertions and deletions in the human reference, people are thinking about using graph approaches to represent the actual reference. So that's something that's quite interesting as well, and maybe graph databases could be used for that as well.
Yeah, it sounds like it could be another interesting use case, wouldn't it?
Mm-hmm.
So so I think Sajid has joined us now. So what should we do? Should we keep on? Do you want to keep on exploring this, or should we let Sajid show us a bit of the import process? Or
yeah, if anyone has any questions, or we can do that. Or
yeah, any biology or Neo4j questions for Nikolas? I'll just watch the chat for a little bit. I think we're good for the moment.
Okay.
Cool. So what should we do? Should we pass over? Do you want to go to the import code on your screen, and then Sajid can explain it? That might be the easiest way for us to do it.
Sure.
Are you able to hear me?
Yeah.
I can hear you.
Great.
We're just going to navigate to the code. So the rough idea of this code is that we've got one of those formats that we saw in the diagram, and then we want to get it in a format that the Neo4j import tool plays nicely with.
Yeah, I guess first, if we summarize what Pheno4J gives us, yeah?
Yeah.
When you check out the code, we've got like a one-shot Maven command. And what this command does is essentially everything, yeah? So in the code base, we've got sample data for all entities, which we've anonymized. And what those Maven commands will do is they'll take that input data, pass it. It'll then dynamically build the import command for the Neo4j bulk import. It'll run that, and then it'll start up your container. And then you can browse to the web UI and then start running queries.
So the example data is in source, isn't it? Test.
Yeah.
Resources. Yeah, here's some sample data. Cool. Do you want to talk about the imports?
Yeah, so in terms of the import, we've designed it so that it can run on like any type of server. So essentially, we're not loading the files into full memory. We read it line by line and then just send it to Google Guava's event bus. And then on that event bus, so essentially it's a pub-subscribe model. You have like multiple listeners. Each one will take a line and then write out the corresponding entity.
Sorry, there's some documentation maybe I should show for this, right? Is it additional documentation, I think, which shows this quite nicely here? Yeah.
Yeah.
That's the one, yeah, sorry.
So do you normally run this, do you run it on your own machine? Do you have like some hardware in one of the cloud platforms, or where's your normal?
So I'm affiliated with UCL, and we are running this on the.
Oh, Nikolas has gone quiet. Oh, what's happened? Nikolas, are you still there? Still browsing, just can't hear anything. Hang on a sec. Maybe I can. So we'll just switch to me presenting while we figure that out. So Sajid, hopefully you can see the Pheno4J again.
Yeah.
Yeah, cool. So we were just going. I guess we were going through. So we've looked already at the diagram at the beginning, and then I've actually tried this, so I can confirm it does indeed work. So that's cool. And then the last thing we got to was this additional documentation down on the end.
Yeah.
And so this one was explaining. So you were talking about the Guava workflow. So where's that? Is that?
So it's being used like inside each of the parsers. So
Okay.
if you want, you can run each one standalone. If you just say doing some testing.
Yeah.
Or yeah, we've got like helper commands in there that you can run all the parsers in one go.
Cool. Yeah, so I think I was looking at it, and each of these is called. I think they're all called sequentially, aren't they, by default? Although they have threading inside them, if I remember.
That's
So
right.
maybe I can. I just search. Which one do you think is the most interesting one for us to look at?
I think maybe annotation parser.
Okay, I just search for that. And so if I just search for annotation parser so you just type T and then you type in annotation parser, and then it will take us. So this is the Python one I think that Nikolas mentioned before. So if I just click the Java one.
Yeah, if you just scroll down.
So tell me when to stop.
Yeah, if we stop here, so you see this block here, create subscribers, yeah?
Yep.
So what this is doing is each of these subscribers is going to take a line and then do something with it, yeah?
So so they are, okay, so they take in an output folder. So is that like a folder on the disk where they're going to write to or something?
That's right, yeah.
Okay. And then they are, this I guess is how you glue together the Guava queue that you mentioned. So are they subscribing to that queue? Is that
That event bus, yeah.
Sorry, the event bus. Yeah. Okay, cool. So you've got lots of different ones. So these for all, what are they for? Are these all different data sources?
So it's for the type of file it's going to write out. So if we say, look at that first one here.
So should I look up this one?
It's not necessary, but yeah, if you want to, yeah.
Oh, okay. Okay.
So yeah, if we look at the name, yeah, gene to generic variant. What that's going to do is going to write out the file for that relationship, yeah?
Yeah.
That's the relationship gene to generic variant. If we go to that list, the next one is generic variant subscriber. That one will write out the generic variant node, yeah?
Yep.
So yeah, each of these files is either a node file or a relationship file.
So you do one file, is it one file per label and one file per relationship type? Is that the structure?
It's one file for a node and one file for the relationships.
Okay, cool.
We've got them separated.
Cool. So then we've got create subscribers there. Then if we come down, so this is now taking presumably this is a wrap around an input file, is it?
That's right. So we've got like a base class, but essentially this is where it's happening, yeah? So we're reading the file in line by line.
Yeah.
In this case, we're reading JSON. So we convert that JSON to a bean, and then that bean is just sent to the event bus.
Cool. And so then when you post it here, one of these will pick it up.
So it'll get sent to all of them.
Oh, okay, good. And so they choose which ones they respond to. Or
yeah.
Like so if maybe we can go to it. This is the gene to genetic variant subscriber. So presumably it's filtering based on these types here, is it? So it works out whether or not it's actually even going to get it.
That's right, yeah.
Cool. And then I guess some of this is in the base class, isn't it? So output, effectively we add it.
So what we have here, if we just look at this example, it's extending something called a set-based subscriber.
Yep.
What happens here is that we have to deal with like duplicates. Yeah, we have like one-to-many relationships in there.
Okay.
So yeah, so we only want to write out the distinct ones. So in there, it's just collecting them into a set, and then at the end, it'll write out the entire set.
Okay. Cool. All right. So and then you basically have something very similar to that for each of the other types of data as well. So that was the annotation powder.
Yeah.
I guess we have the same thing for the other ones as well.
Yeah. The structure is all the same. You just set up a load of subscribers.
Yeah.
And then you read line by line, send it to the bus. That's it.
So I don't remember. Are there any examples of what the output files look like checked into the repository somewhere?
The output we don't have. If you run the command, it'll generate it for you.
Oh, okay, could give it a try.
Yeah, yeah.
So that would be just if I do this bit, right? So let me just go to. I'll just go to my command line. I'll zoom in a bit. Cool. So if I just paste that in. So this is what it looks like when it's running. Okay, so we're just doing a bit of compilation. Now it's processing. So I guess it's going through the files now, isn't it?
Yeah, well, we can't see the screen.
Oh, that's. Hang on a minute. Let me see if I can figure out how to fix that. So if I share screen, I'll just do entire screen. Now I guess you can?
Yeah.
Okay, sorry, I've skipped through all the workings. That was a bit of cheating there. But so if I scroll back to where we were before, so we were right up here. Yeah, so we were processing these lines. So we did 280,000 lines, and then we can see it's writing out some files. So that's the gene to term one, and then it goes through the others. And then once it's got the CSV files, so we kind of get done with the CSV files, it then generates a command that you should call with the Neo4j import tool. So that's here. So this is what the Neo4j import tool looks like. So you tell it where should it create the database, and then you tell it where all the nodes and relationships are coming from. So we've got lots of different node files here, and then the same. And then it's the same for the relationships. And then finally, we can go and create the database. So maybe I can open up some of these files, and we can have a quick look what they look like. So this is an example of a header file, and maybe I'll let you talk through it.
Yeah, so it's good that we pick generic variant because we hold very large number of attributes for it.
Yeah.
If you just look at the first row and go like a few words in, you see that we've also typed these columns, yeah? So we've got integers in there.
Yeah, so for like strand.
Yeah.
Cool, so this is the header. So this is indicating what's going to be in the CSV files that are going to be specified next. If we go to now genetic variant.csv, is it right? Oh, no.
It's hyphen and then the name of the parser that generated it.
And maybe annotation. Yeah, so we go to annotation parser. So now we just have all this is the data for each row. So I guess this is one row.
Yeah.
And then they all map up to what's in the header file.
That's right. Yeah.
Oh, cool. So it's mostly numbers. I see a inbreeding maybe coefficient hovering at the end of the first line.
Yeah.
Cool. And then we could see further up the screen that there were lots of different files. And then when the Neo4j import command processes it, it kind of goes through those files and it creates the nodes first. And then the next thing you'll see is there's a dense node section. So that's any nodes that have more than 50 relationships coming from them. There's a special way that those relationships get stored. Then we're kind of just going through the different relationships after that. Cool. I don't know. I'm not sure how much data we loaded. Maybe it says at the end. So we did. Oh, here we go. So we've got. So this is a. Did you say this is like a? Is this a real data set but anonymized, or is it a generated one?
No, it's a very cut down amount. Yeah.
Okay. So we've got just over 250,000 nodes. We've got just over 500,000 relationships, and then a similar number of properties. And we've managed to load that. So it only took 10 seconds. So that's pretty cool.
Yeah, it's what we liked about the bulk import. That it is very fast. In the production system, we have 20 million nodes.
Okay.
And about 600 million relationships. And yeah, in terms of volume, it's taking up only about 20 gig of disk space. Cool. So yeah, we also got like very good scalability out of it.
Yeah, cool. And so does it update very often, the graph?
So it's something Nikolas is working on now. So in one go, we can build up the whole database, but sometimes they have samples coming in where they just want to load in a small amount of data.
Okay.
And there, yeah, he's looking into the load CSV cipher command.
Oh, cool. Okay. Nikolas, are you back yet? Maybe not.
I think maybe one thing we should also discover is what we see at the end of the log there.
Oh, sorry. Hang on. I'll share my my screen again. Can you see my screen again?
Yeah.
So where do you want me to go? Just down a bit?
Yeah, so that last block there, yeah?
Yeah.
What we see is it's creating constraints and indexes. So we also do that programmatically.
Okay, cool. So that's from the Java code that we looked at before.
Yeah, yeah. So yeah, so the Pheno4J solution with that one Maven command, will do everything for you. Yeah. Then
Cool.
if you want, you can look into each process.
Yeah. So I think I skipped one of the steps, didn't I? I didn't do the run Neo4j bit. So I've got the data. I think I've got the database there if I want to use it, but I didn't actually launch it. So I think so. Nikolas is saying his microphone's not working, I think. So Nikolas, are you back again? Maybe not the microphone, not yet. Otherwise, Sajid, do you have the Phenopolis? I think the next thing we were going to look at was the Phenopolis web app. Do you have that on?
The Phenopolis I don't have. There would need Nikolas.
Okay. Maybe you can share your screen, and then would you be able to talk through it?
The Phenopolis, I actually don't have access to it here.
Yeah, but but Nikolas could share it maybe, and then if you're able to talk through it, that might. Yeah. So not quite optimal, but maybe slightly better. Okay, so that right, here we go. Here's the Phenopolis application.
Yeah. So in terms of knowledge I have, it's around on the server side. So what we're doing on the server side is it's all Python based, and the Python is just running Cypher queries, and those Cypher queries is JSON based, which gets returned to the Python GUI, and it's just rendering those JSONs.
And so who would be the user of the Phenopolis app? Actually, if I go back a little bit, like, is it used by researchers or analysts, or who would be browsing this API or this interface even?
So Phenopolis, yeah, if you go on there's a about page, and there, yeah, you'll see it's used by a list of universities. Yeah.
Okay, cool.
Can you hear me?
Oh yeah, Nikolas is back again. Cool.
Oh, good. Okay.
Perfect timing, Nick.
Okay. Yeah, perfect. Okay. So, yeah, maybe I'll go for this because that's more on the research side rather than the implementation. So you want to give you an overview of how this is used. So yeah, so what we're browsing here is a subset of the patients, and I just opened one of them here in this tab, and what you see here is we were talking about the variants. So we're only looking at rare variants here. So in fact, in reality, there's 100,000 of these variants, but here we're only looking at the rare ones. So this is, I should say that we haven't finished integrating the new Pheno4J database with this. So this is currently still using the Mongo database. But the idea is once we've got the Pheno4J linked up, the graph database, we'll be able to display many more of these variants, and we'll also be able to give the user the possibility of choosing what variants they want to see in what genes and at what allele frequency and so on. So if you go to, this table represents the variants here, and here are the gene names. So if I go to a gene, so this is a demo account. I'll log into them. Maybe I'll give you a view of them. So the demo account is restricted in the genes you can see. I think actually, can you see this gene? Yes, you can only see a few genes. So you can see the DRAM2 gene. So there you go. So this list of variants here is the list of variants in that gene. So if you remember that when I showed you the graph database here, we were looking at the there. So for instance, the number of variants attached to that gene. So that's what these rows these rows are here in the table. Sorry, 1 second. Yeah, here you go. And then there's other features here, but it might be a bit complicated to explain this. Well, I can explain this actually, this bit here. What you're seeing here is I was referring to the clinical features early on, and in particular to the human phenotype ontology, where what you're seeing here is a subset of this ontology. So we're only displaying the clinical features seen in patients which have mutations in this gene. So it's kind of trying to link the gene to the clinical features, basically, by looking only at the mutations in patients which have in the mutations in these genes. It's getting a bit complicated to explain these levels of indirection, but yeah, here we're not displaying the patient IDs because it's the demo account, but in the admin login, which I can't really show you in the demo, but yeah,
probably best not to do that on YouTube.
there's no name. Yeah, there's no names of how many patients, but still, it's better not to show it. So yeah, so that's the gene view that I showed you, the person view, and and the other view is if we go to a term. So this is a HPO term, so a clinical feature term, and this just shows you all the people which have this clinical feature. And so there you're seeing the term to person relationship effectively, and these are all other terms, so I could go and explore these other terms as well, and so on. And one thing that's interesting we would like to show as well is maybe how many people have each of these terms. So it'll be more obvious which terms are rare ones and which are common ones, because these are all clinical terms which might not mean much to you, but some of them are quite rare and so informative, whereas others are quite common and less less pertinent.
I recognize asthma.
Yep.
That's the limitations of my knowledge.
Yeah, so if you click on asthma, we have 2 patients with asthma.
And then those are the other things that they...
Exactly, yeah.
Mm. Wow, cool. We've got a question from the chat. I'm not sure whether I'm going to completely get it right, but I'll try. So this is from Fernando Izquierdo. Says asking, can users write their own queries to explore the graph via Phenopolis, or does it rely on predefined queries / API?
Yeah, so we have we're using Ajax. So we're using Ajax endpoints, so JSON endpoints. So if you go, I can show you the code basically. So this is the dev branch on which we are big. So the master branch at the moment in Phenopolis is using the old database, but this is the dev branch, and we started using Cypher here. So we've got like this, which is is being called by this endpoint here. So yeah, but at the moment, I guess if you wanted to do your manual exploration, you would do it maybe through the Neo4j interface. But obviously, we can't just let anyone look at all the data. You need to be sort of working as part of the team, or if that's the question. I'm not sure if I've answered it.
Cool. Yeah, I guess I can wait for Fernando to indicate whether we've covered what he was asking. Cool. So I guess we're kind of 49 minutes in. Is there anything else? So I guess we talked about, so your next thing you're working on is the Phenopolis API, and you said it was currently using Mongo, and then you're going to try and put Neo in there. Have you already started doing that, or it's in progress, or what's the?
So that's the the Dev1 branch on the Phenopolis GitHub, and in fact we're very happy if anyone wants to contribute to this to this project. So the idea is this website can be used for anyone in doing the same sort of research as we're doing, and we want to. There's some private commercial alternatives at the moment, but this is we want to have a sort of open source version of this, so that people can browse their genetic data easily.
So would it be possible if you had your own genetic data, you could maybe import yourself into the Pheno4J? Is that
I do.
Oh, you do? Oh, wow. Okay. Already ahead of me.
I'm in the database already. Yes.
Oh, wow. Cool.
Yeah.
So what would you do? How would you get your data? Is there like some services that you can use?
Yes, there are some services, or
Okay.
you can know the right people as well. But yeah, there's well, you might have heard of 23andMe and
Yeah, I've heard of that one.
yeah, so
Something similar to that.
but there's there's OpenSNP as well, which is a website where people can upload their genetic data, and then it'll give you, but it won't necessarily let you browse in the same way all the data. I mean, our website's really for clinical cases, so we have very detailed phenotypes which use human phenotype ontology, the HPO terms.
Yeah.
And we are really looking for new, it's a discovery platform as well, because we're trying what I showed you in the gene page, which was, can't remember where I have it now. I closed it, I'll open it again. But yeah, it shows you the relationships between people and sorry, the relationship between genes and phenotypes. So, is it TTR5? Yeah, we're showing like patients which have a certain clinical feature, which have more mutations in that gene than than expected by chance, basically. So here, for instance, we're showing that this gene, according to our database, is linked to macular dystrophy. So we've got a p-value there. So yeah, it is a discovery platform as well.
So as a p-value, that's like from statistics land.
Yeah, it's
Okay.
it's just a Fisher test. The idea is, sorry, it's not great to click on it on the demo mode, but here, if you see, you have an expected frequency and an observed frequency.
Oh, yeah, okay.
Number of people, basically the idea is number of people which have this clinical feature in our database and don't have a variant in its gene, versus number of people which have that clinical feature and have mutations in this gene, basically.
And then the p-value is sort of saying whether or not how likely that is to have happened by chance as compared to there's an actual difference.
Yes, yeah, exactly. Okay. And what you have here, actually, these sliders, which would work nicely in using the graph database, is how we filter the variants out. So if we have more stringent filters, then we'll get less variance for, and so that tells you a number of matching variants. There you go. So if I increase that to, say, 10, oh no, sorry, that's not the right thing. So if I increase this to 10, sorry. So yeah, that's one. The top one, the bottom one is the score of how damaging the variant is, and the top one, is the frequency, basically. So if I set a lower frequency here and a higher frequency and a lower CAT score, I get more variance through. But if I increase the CAT score, I'll get less variance through. So you have to. We are also investigating how to set these thresholds optimally. But yeah, basically here we've got 13 variants through, and if you click at the top, you see here the number of patients with mutation, which have this HPO term.
Yeah, cool. And so if people wanted to contribute to the project, where would you suggest they start looking?
So we've got a nice, what we think is quite easy way of starting is to run. We have a script to do a quick installation. So we've got here quick install for coders. So this at the moment is using the Mongo database, but we're transferring it to using the graph database, to use Neo4j. But yeah, this script basically has all the commands in order to set it up with some demo data.
Oh, cool. Yeah. Okay.
So yeah, so we're going to write that.
So we can share the link for that as well.
Great. Yeah.
Cool. So I guess we've probably covered most stuff. Is there anything else we haven't covered that you want to?
No, I mean, you can look at these. So Phenopolis is published, so you can look it up on. I don't know if there's any academics attending this chat, but you can look it up on PubMed, and also Pheno4J. So Pheno4J is actually still in preprint, but it will be in bioinformatics journal soon, so you can read the paper if you want to know more.
Cool. Well, I reckon we can wrap up. So thanks Nikolas and Sajid for talking us through Pheno4J.
Thank you.
And thanks everyone for joining us. So next week we're doing how to build an OGM for Node.js. So if you're free this time next week, I'll see you for that one. Otherwise, yeah, thanks again, Nikolas and Sajid.
Great. Thanks a
Bye,
lot.
everyone.
Thanks.
Take care. Thank you. All right.